Good morning, one and all. This is Satyajit Beura from Department of Bioscience and Biotechnology, Indian Institute of Technology, Kharagpur. Today, I am presenting my part of my research work that illustrating metabolic interaction in the gut microbiota of type 2 diabetic patient. So, before going into the topic, I want to discuss what is human gut microbiota. Human gut microbiota is the composition of microorganisms that includes bacteria, archaea, and microscopic eukaryotes. These includes nearly 300 to 1000 different species, and most of the microorganism, around 99% of the microorganism are belongs to 30 to 40 species. And what are the function of these microorganisms? So mostly these microorganisms maintain a symbiotic relation with the human gut environment, so they help in the digestion of the complex products. But in case of the imbalance in the microbial population, that disturbs the human biochemical pathways that leads to different diseases. That disease includes cardiovascular disease, arthritis, inflammatory bowel disease, obesity, cancer, and type 2 diabetes. Here we are emphasizing on type 2 diabetes. So these are some list of microorganisms that are highly altered in case of type 2 diabetes. There are abundance of those microorganisms are highly altered in case of type 2 diabetes. That includes Prevotella acropli, Lactobacillus rhamnosus, Mithraeus smithii, and followed by some other microorganisms. And these microorganisms are basically belongs to complex carbohydrate degrader, energy harvesting microorganisms, certain producing microorganism. From different literature survey, we have also found that these microorganisms are crucial for human health, and mostly these are found in case of type 2 diabetes. It is also found in different studies the abundance of these microorganisms are altered. So based on the different literature study and our data analysis, we have identified these three microorganisms, that is Prevotella copri, Lactobacillus rhamnosus, and Mithraeus smithii for our study. So for our study, we are reconstructed the genome scale metabolic model of all these three microorganisms, followed by the community model, and analyze the how they are altered, their metabolic behavior altered in different conditions, that is healthy and type 2 diabetes. The first step, this is genome scale metabolic model. Genome scale metabolic model is a in silico model that designed computationally, which shows the total metabolic activity of one microorganisms. So for that, the whole genome sequence of one microorganism is considered, and that is been annotated using different web servers. And after annotation, you can see that the whole genome is assigned with the respective genes and then the respective proteins and the respective reactions. These reactions are get connected to construct a genome scale metabolic model. But this genome scale metabolic model is considered as a draft model, so that we have curated these models by using different data sources, web sources, and also we follow the literature, so that we get the reaction, appropriate reaction to fill the gap present in the reaction or present in the model. And also we perform the mass balance analysis, charge balance analysis, and all those things to get a perfect genome scale metabolic model for one microorganism. Once we got the model, then we want to check the model is well enough for our work or not, so that we followed the sensitivity analysis. The sensitivity analysis, what we did, we simply take all the precursor molecules, and we take all the reactant of the biomass reaction and change the concentration by plus minus 10 to 15%. And we check that by changing the concentration of the precursor molecule, the growth rate of the microorganism is not changed that much. So its growth rate is changing 2 to 3% maximum, and these changes are mostly due to the amino acids and nucleotides. So this indicates the models are highly curated, well connected, and robust in nature, so that we can easily get into the next step. We can integrate the model to reconstruct the community metabolic model. For the genome scale community metabolic model, all the three models are integrated to construct the community model. But this community model is known as a generalized model. So make it got specific. What we did, we integrate the coefficient of those microorganisms. That means the abundance of those microorganisms as a coefficient of this of the reaction reactant, so that we can make it condition specific or got microbiota specific. So this is for the healthy community, and this is for the type 2 diabetic community. Next, for the analysis, we perform the flux variability analysis. So flux variability analysis shows the range of flux through which point flux can be flow. So we perform the flux variability analysis for healthy and type 2 diabetes. Here you can see that the range of the flux in the healthy and range of the flux in type 2 diabetes are distinctly all changed. So more specifically, to get it more clear idea, so I plotted this heat map. So you can see that for every pathways how the range of fluxes are changing. So for example, I am showing here a reaction in the carbohydrate metabolism. The maximum flux range is 0.99 in healthy, but when it is getting entered into the type 2 diabetes, you can see that it's getting down to 0.01. If the range is reducing, that means the point flux value also going to be reduced. So based on that, we have performed the mapped all the point flux value. Here we mapped the point flux value, and here we got that all the three microorganisms are helping each other to grow in the gut environment. They are also consuming some metabolites like nucleotides, amino acids, certain fatty acid, and produce some essential components like also like amino acids and vitamins and all that. But in the case of healthy and type 2 diabetes, we have changed some difference. The major one is the glucose. The intake of glucose molecule is very high in the healthy community, but when you are comparing this with the glucose consumption in type 2 diabetes, you can see that it is drastically reduced. That means when the consumption of glucose molecule is getting reduced by the gut environment, microbial community, that leads to retention of glucose molecule in the blood serum. That leads to the hyperglycemia. Addition to that, you can also see that bicarbonate concentration is also getting reduced. That means the production of bicarbonate. It is also known that the bicarbonate concentration is decreasing with respect to the proceeding with the type 2 diabetes disease. And also you can see that here easily the certain fatty acid that includes acetate and a few format, these molecules are also reducing, and this is majorly known as, these molecules are known as the they help to maintain the mucosal layer. Reduction of these compounds hampered the mucosal layer and the absorption of the nutrients. So for this whole study, we have identified that some crucial molecules are there like format, acetate, bicarbonate, and some amino acid get exchanged within the community. And the major thing is you can see that in the flux variety analysis, amino acid, vitamin biosynthesis, carbohydrate metabolism, all these things are getting down, and the glucose consumption by the community is getting reduced, which leads to the retention of more glucose in the type 2 diabetic patient, which leads to the hyperglycemia. And along with the glucose molecules, some other molecules like nucleotide, vitamin B5, cysteine, phenylalanine, and format, other molecules are also getting affected. So these are some references of my study, and I would like to thank the Department of Bioscience and Biotechnology, and School of Energy Science, Indian Institute of Technology, Kharagpur, Ministry of Education for the fellowship, and ISCB for organizing the conference. Thank you.